Okay, so as a part of Unacademy Current Affairs Navigator, today we will be discussing a few current affairs from the duration of 25th October to 17th November. And for each current affairs article, we will be discussing a multiple choice question so that you will get an idea about what kind of questions you can expect from that topic. So the first topic we are discussing is caterpillar fungus can slow down the growth of the cancer cells. So let us first try to understand what is cancer. Okay, first of all, what is cancer? Cancer is nothing but unregulated growth of cells. Unregulated growth of cells, that is excess growth of the cells. And in our body we have two mechanisms. One is cell division, and second one is apoptosis. Apoptosis. All of you just memorize the term apoptosis. Apoptosis is also called as the programmed cell death. Apoptosis is also called as programmed cell death. So, please answer in the comments by which process the cell number increases in our body, cell division or apoptosis. By which process the cell number increases in the body. Because of the cell division, the cell number increases, right? Because of the apoptosis, the cell number decreases. Because it is programmed cell death, the cell number decreases. So, answer this question. Cancer occurs because of the increase in the cell division or increase in the apoptosis. Cancer occurs because of the increase in the cell division or increase in the apoptosis. Cancer occurs because of the increase in the cell division and decrease in the apoptosis. So logically, when does the cell number increase? When the cell division increases or the cells do not die, the death is not happening properly. In both the cases, the number of cells in our body increase, so that leads to cancer. This is a little bit of background about what is cancer. Fine. Now let us try to understand what is this caterpillar fungus. There is one moth like a butterfly called as ghost moth. Use the name of the moth, please. Use the name of that butterfly once. Answer that. Ghost moth. This butterfly, when it is in the larval stage, that is in the early stages of its development, we also call that stage as caterpillar. This ghost moth, when it is in the caterpillar stage, it is sometimes infected by a fungus. Just like we get fungal infection sometimes, like black fungus infection and all. This caterpillar also will get some fungal infection. That fungus is called as caterpillar fungus. It is called caterpillar fungus because it is infecting the caterpillars of the ghost moth. The scientific name of this caterpillar fungus is Ophiocordyceps sinensis. Ophiocordyceps sinensis. That is the scientific name of that. This caterpillar moth is producing a particular chemical called as cordycepin. Use the name once. Message me the term of that chemical, cordycepin. I repeat, ghost moth, it's caterpillar, sometimes infected by a fungus called caterpillar fungus. Name of that fungus is Ophiocordyceps sinensis. This fungus will release a certain chemical called as cordycepin. This cordycepin is having a capacity to cure the cancer. It interrupts the cell growth signals. So how does the cordycepin decrease the cancer? How can the cordycepin cure the cancer? by interrupting the cell growth signals. Cell growth signal means what? Just like we have a fan switch, light switch like that, we have a switch for the cell division also. Okay, this cordycepin is switching off that cell division process. The cordycepin will switch off the cell division process so that the cells will stop dividing. So when the cells stop divide, can we say that the cancer is getting controlled or cured? The cure of the cancer is nothing but decrease in the cell division. Okay, so that is how the cordycepin decreases. So ghost moth, its caterpillars infected by caterpillar fungus called as Ophiocordyceps sinensis, it will release a chemical called as cordycepin, and cordycepin can cure the cancer. How can it cure the cancer? By switching off the cell growth signals. Okay, so that is a summary of the news article. Now let us try to answer this multiple choice question. Take some 30 to 40 seconds time and try to answer this question. You can send me the answers in the message in the comment section. Cordycepin interrupts cell growth signals to control the cancer. Cordycepin activates apoptosis to control the cancer. Cordycepin is a chemical synthesized by caterpillar fungus. Which of the above statements are correct? What is the correct answer? Everybody please. I'll wait for 10 more seconds. Okay. The answer is B. 1 and 3 is the right answer. Cordycepin interrupts the cell growth signals to control the cancer. Yes, it is not interrupting, it is not activating the apoptosis. I mean, in general, if apoptosis increases, cancer can be cured. Your logic is correct. Some of you answered all the above. The logic you applied is correct. If the apoptosis is activated, you can control the cancer. That is correct. But that is not the mechanism of this cordycepin. Is it clear? The mechanism by which cordycepin is controlling the cancer is by switching off the cell division, not by activating the apoptosis. I hope you all got the point. If you activate apoptosis, cancer cures. But cordycepin is not activating the apoptosis. So this is not correct. And cordycepin is a chemical synthesized by the caterpillar fungus Ophiocordyceps sinensis. Correct. 1 and 3 are correct answers, correct statements. So the answer is 1 and 3 only. Right? Okay, moving on to the next article. Why Cambodia's H5N1 reassortant virus needs close monitoring? Okay. First of all, message me the term in the comments. What is that virus? Reassortant virus. Use the name once. Reassortant virus. Is it clear? Reassortant virus can also be called as recombinant virus. Recombinant virus. Both mean the same. Reassortant virus or recombinant virus both mean the same. What is recombinant virus? Let us suppose there are two clades of H5N1. Okay. What do you mean by clades? The word clade means what? Like strains. The famous example in COVID-19, we have seen the strains like Omicron strain, Beta strain like that. The strains of H5N1 are called as clades. What are the two clades that you can see here? 2.3.2.1c, 2.3.4.4b. These are the names of two clades of H5N1. Is it clear? Let us suppose Geeta got infected by 2.3.2.1c and Sita got infected by 2.3.4.4b. Is it clear? After some time, Sita also got infected by this virus. I hope it is clear. Same person infected by two different clades of the H5N1. Clear for all of you? Same person infected by two different strains or clades of the H5N1. Then what happens here? The two clades, the first clade, okay, these two clades will undergo some recombination. Recombination. It means what? They combine with each other. Okay, they kind of a hybrid. They both will exchange the genetic material. They combine with each other. They form a recombination. Due to this recombination, you will get a new virus, which is like a hybrid between or a recombinant between these two clades. I hope it is clear for all of you. It's a recombinant between the two clades. Right? Clear? Okay. So what are the two clades? 2.3.2.1, 2.3.4.4b. These are the two clades. The same person got infected by both of them. They both underwent recombination. A new combination between the two created the reassortment or recombination and a reassortant virus. The novel reassortant virus, novel means new. The new recombinant virus caused deaths as of now. But there is no evidence of human to human transition. Is it clear? It is basically H5N1. Is it human influenza or a bird flu? H5N1 is human influenza or bird flu? It is basically bird flu. So we are getting the infection from the birds. Birds to humans, some infections are there. Some deaths also occurred. But one human being to another human being transmission is not yet reported. Is it clear? So, do we have human cases of bird flu? Did some human beings get bird flu? And did some human beings die with this bird flu also? Yes, they died. They got the infection, they died. But is human to human transmission of this bird flu reported in any case? No, human to human transmission is not reported. Okay, so what they have done is they have taken each human case. They have taken each human case and they have done a collaborative One Health investigation. Did you hear about this One Health approach? Can anybody answer in the comments what is One Health approach? One Health approach is a collaborative approach towards diseases where we consider, okay, yes, Saurabh, correct, where we consider humans, okay, animals. Animals means it will include birds or insects, all animals, and also the environment. Is it clear? It is a holistic approach. Clear for all of you? What is One Health approach? In the One Health approach, we do not study the aspects of the diseases only in the case of human beings. We study the interaction between humans, animals and environment. Is it clear? For all kinds of organisms. Any infection comes, we don't study this only in the human beings. We study this in the human beings, in the animals and also environment. Interaction between these things. That means how the animals are causing diseases in the humans. How the environment is causing diseases in the humans. The holistic approach between all the three components together is called as One Health approach. Very good, Saurabh. I hope it is clear for all of you. Interaction between humans, animals, environment. How the environment will cause diseases in the humans. How the animals will cause diseases in the human beings. What is the interaction between all these things? That kind of an approach is called as One Health approach. Okay. So for each human case, they are doing a collaborative One Health investigation. Okay. It was conducted, which was able to link the human cases to the infected poultry. Means what they have done is, they have taken the infected poultry, that is bird, and isolated the H5N1, did the DNA sequencing, and they have taken the human sample from the infected person, isolated the H5N1 and did the DNA sequencing. Interestingly, the DNA sequence of both the H5N1 is matching. It means human is getting this infection from that bird. I hope it is clear. I repeat the process. Okay, take the sample from the infected human being. Isolate the bird flu virus H5N1. Do the DNA sequencing. Then collect the sample from the infected bird. Isolate the H5N1. Do the DNA sequencing. Compare both the DNAs. They are matching. It means human being got this virus from another human being or the bird. Answer please. The DNA is matching means the human being got this infection from another human being or from the bird. How did we know? Because our DNA sequence, the H5N1 DNA sequence in the humans is matching with that of the birds. Okay, through the rapid whole genome sequencing. Why this is called One Health investigation? Because they are comparing the H5N1 in the human beings and the birds, not exclusively human beings. They are not studying about the H5N1 exclusively in the human beings. Is it clear? They are studying about H5N1 in human beings and the birds. Clear? Holistic comparison. Okay. Therefore, it is called One Health investigation. Researchers detected PB2627K mutation. Okay. Use this word once. PB2627K mutation. What do you mean by this? There is one gene called as PB2 gene. Is it clear? PB2 gene. This gene will produce a protein called as PB2 protein. It will produce PB2 protein. Okay. This protein helps in replication and transcription of the virus. Is it clear? This protein helps in replication and transcription of the virus. I hope it is clear. A gene called as PB2 gene. The gene produces a protein called as PB2 protein. This protein is critical for the replication and transcription of the viral RNA. Replication means viral RNA getting copied, another copy. Okay, and transcription of the viral RNA. That means expression of that particular genes. Okay, it is very crucial for that. So, in this gene, in the PB2 protein, I hope you all know that proteins are made up of amino acids, isn't it? Proteins are made of amino acids, right? Amino acids are the building blocks of the proteins. Let us suppose this is a chain of amino acids. Let us assume each ball is an amino acid and this complete chain is a protein. Which protein it is? PB2. Is it clear? Which protein it is? PB2. Okay. Now let us suppose 1, 2, 3 and so on. Let us suppose that it is the 627th amino acid. Did you understand? This is the 627th amino acid in the PB2 protein. In this amino acid, originally there will be, okay, the position 627 in the PB2 protein where lysine, okay, originally there is some amino acid called as glutamic acid. There is one amino acid called as glutamic acid. Glutamic acid, okay, symbol is E. Okay, tell me in the 627th position of PB2 protein, which amino acid is present originally? in the 627th position of the PB2 protein, which amino acid is present originally? Glutamic acid. Glutamic acid is represented by the letter E. Okay, then because of a mutation, the glutamic acid is replaced by lysine. Lysine replaces the glutamic acid. This will get converted into lysine. Lysine is represented by the letter K. Is it clear? Okay, I repeat, PB2 is a protein. Whose protein is it? PB2 is a protein in whom? Human being or the bird or the H5N1. It is a protein in the H5N1. PB2 is a protein in H5N1. It is responsible for its replication and transcription. Okay. Then in that protein 627th position, originally there is glutamic acid. Because of a mutation, this is converted into lysine K. Therefore, this mutation is called as PB2 627K mutation. I hope you understood this. PB2 protein 627th position. Okay, you have lysine K. Therefore, it is called as PB2 627K mutation. We have this particular mutation in the recombinant virus. We have this mutation in the recombinant virus. I hope it is clear for all of you. Recombinant virus is also called as reassortant virus. Reassortant virus. Tell me which virus is having this PB2 627K mutation? The recombinant virus. Okay. Survival of the H5N1 inside the humans. Okay, Roger. It is responsible for PB2 protein is very important for the H5N1 to survive in the human beings and also to replicate in the human beings. That protein's 627th amino acid glutamic acid is replaced by lysine. Therefore, we are calling the mutation as PB2 627K. We have this mutation in which virus? We have this mutation in the reassortant virus. Is it clear? Fine. What is the impact of this virus? What is the impact of this virus? Mutation. The impact of this mutation is enhanced mammalian adaptation, means the virus having this mutation is better adapted to mammals, not only the birds. It can better adjust. Adaptation means what? Adjustment. It can better adapt in the mammals, not only the birds. Is it clear? Okay. Because of this, the virus having this mutation, that is that recombinant virus having this mutation, can better adjust to the mammals. And also it will increase airborne transmission. That is, when you sneeze or cough through the air, that virus can easily spread. I hope it is clear. The virus having this mutation can better adjust in the mammals like human beings, and also it can make the virus easily spread through the air. Is it clear? So it means, can this mutation make this virus a zoonotic virus? This mutation will increase the zoonotic potential. Fine. What do you mean by this word zoonosis? Zoonotic diseases. Zoonotic diseases means the diseases that spill over, that spill from animal to the human being, like rabies, coronaviruses, bird flu. All these are the zoonotic viruses. How this mutation is responsible for zoonotic? How the mutation can make H5N1 zoonotic? Because due to this mutation, the virus can better adjust in the mammals and can more efficiently undergo transmission through the air. So therefore, this mutation is having the potential to make the virus a zoonotic virus. Okay, fine. This is the summary of the article. This article published in the Hindu, 10th November 2024. Now, participate in the poll. I don't have a poll, but like go through this question and try to answer. Consider the following statements. One Health is focusing particularly on the virus impact on the humans. Particularly on the virus impact on the humans. Novel recombinant virus combines gene segments from the clade 2.3.2.1c and 2.3.4.4b. The PB2-627K mutation for mammalian adaptation and airborne transmission is found in the new recombinant virus. PB2-627K mutation can make the bird flu a zoonotic infection. Yes, very good, very good. Thank you so much for the active participation. The correct answer is D. One is wrong. Okay, these three are correct. One Health is focusing not particularly on the virus impact on human beings. Humans plus animals plus environment. A holistic approach is One Health. And recombinant virus combines the gene segments from the two clades. Yes, correct. That is the reason why we call it as reassortant virus or the recombinant virus. That recombinant virus is having this mutation. Because of that only, the recombinant virus is capable of easily adjusting in the mammals. Correct. Fourth statement, PB2-627K mutation can make bird flu a zoonotic infection. Yes, because of this mutation, easily the virus can spill over from the birds to the human beings. Okay, so the correct answer is 2, 3, 4. Very good. Thank you so much for active participation. Moving on to the third topic in today's session. Can we proceed? Layer of graphene oxide halts frost formation on the flat areas. Layers of graphene oxide halts frost formation on the flat areas. So first of all, okay, what is graphene? Okay, tell me what you know about graphene. Is it a nanomaterial? Is it a nanomaterial? Yes. It is made up of what? Graphene is made up of carbon, and it is an allotrope of carbon, isn't it? Graphene is an allotrope of carbon. What do you mean by allotrope? Is allotrope isotope same? Is allotrope and isotope same? No, allotropes and isotopes are not the same. Allotropes means same carbon, just differ in the spatial arrangement. Differ in the spatial arrangement of carbon atoms. Is it clear? No chemical difference. No difference in the atomic number. No difference in the mass number. No difference in the position in the periodic table. The same carbon. But just there is difference in the spatial arrangement of the carbons. Such different structures are called allotropes. Okay. Look at here. This is graphene sheet. Okay. If you observe the graphene sheet, graphene is... It is an allotrope of carbon and it is a flat sheet, isn't it? It's a flat sheet of carbon. Okay, it looks like a flat paper. Okay, it's like a flat sheet of carbon, and its thickness is just 1 atom thick. The thickness of this graphene is just 1 atom of carbon. It means it is a very thin sheet of carbon units. Is it clear? It is a very thin sheet of carbon units. Right. So, can you guess and tell me, is it a 2-dimensional structure or a 3-dimensional structure? Is graphene 2-dimensional or 3-dimensional? graphene is two-dimensional because it will have length. Okay, just look at this paper. This paper is having length. Breadth is also there. Length is there. Breadth is there. But thickness is negligible. One atom thickness. So it means depth is not there. Did you understand? Length is there. Breadth is there. Depth is not there because it is only one atom thickness. Is it clear? I mean, this is negligible thickness. Therefore, it is having only two dimensions. Length is there, breadth is there, but not the depth or thickness. I hope it is clear. So, two-dimensional flat sheet of carbons arranged in the form of hexagons like this. Hexagon. Carbons are arranged like this. I hope graphene is clear. Next, let us try to understand about fullerenes. Use the word once. Fullerenes. What are fullerenes? Okay, look at here. This is a fullerene. This is a fullerene. It means if I take this particular sheet, if it is flat like this, then it is called graphene. Then if I roll it up like this, then it is called as fullerene. It means any configuration other than the flat sheet, any structure other than the flat sheet. It can be a tube like this, or it can be a ball like this. It can be a ball like this. Is it clear? And they both are three-dimensional. I mean, fullerenes are three-dimensional nanomaterial. All of them are nanomaterials. Is it clear? We call this as buckyball. This is called as buckyball or Buckminster fullerene. And we call this as carbon nanotubes. Carbon nanotubes. Is it clear for all of you? Right. So what is there in the newspaper article? 3rd November 2024 Hindu newspaper. Okay, researchers discovered that, okay, the tweaking means what? Making some modifications. Okay, tweaking the texture of any surface and adding a thin layer of graphene oxide to it. It means if I have a surface, fullerenes are three-dimensional. Okay, they will have length, breadth and depth also. For example, it is a fullerene. It is having length, it is having breadth, it is also having the depth. It's a three-dimensional structure. Okay, right. Graphene is a two-dimensional structure. Let us suppose I have a surface and I don't want frost, that is snow or ice, to deposit on this surface. So what they discovered is, if you give a graphene coating on this, if you coat the surface with graphene, okay, then what happens? If you coat the surface with graphene, it will remain frost free. It will prevent, okay, the frost from forming on the surface for 1 week to 10 days, even longer. This is the news article. It is 1000 times longer than the current state of the art anti-frosting surfaces. State of the art means advanced. So currently, whatever advanced techniques we are using to prevent the frosting, this graphene coating is 1000 times longer duration. It can prevent the frosting. Is it clear? Okay. It can prevent the frosting 1000 times longer than the currently used techniques. The new scalable surface design is also resistant to cracks, scratches and contamination. Not only frost free, the surface also will become resistant to cracks, scratches and contamination. So this is the news. News is over. Okay, and we have seen some extra content about graphenes, fullerenes, carbon nanotubes, buckyballs, all those things. Now, okay, go through this question. The third question in today's class. Okay, read the statements carefully and give the answer. Graphene is single atom thick flat sheet of carbon. one of the allotropes of carbon which can be kept under the category called fullerenes. Yes, if graphene is rolled up, then it is called fullerene. Marley Princess, correct. If the graphene is rolled up, it can be called as fullerene. Graphene is a three-dimensional nanomaterial. Graphene is stronger than steel and more conductive than copper. Stronger than steel and more conductive than copper. Okay, let us check the statements. Graphene is single atom thick flat sheet of carbon atoms arranged in hexagons, correct. Graphene is one of the allotropes of carbon, correct. But can we keep it under the fullerenes category? Can we keep it under the fullerenes category? Yes, can we keep it under the fullerenes category? No. Because fullerenes are like this. This is fullerene, this is fullerene. Okay. But graphene is a flat sheet. So therefore, it is an allotrope of carbon, correct. But it cannot be kept under the category of fullerenes. So this is a wrong statement. Graphene is a three-dimensional nanomaterial. No, it has only two dimensions. Length is there, breadth is there. But the depth or the thickness is negligible, one atom thickness. So this is also wrong. It is not three-dimensional. Graphene is stronger than the steel. Yes, it is 200 times stronger than the steel and more conductive than the copper. So therefore, we can also use them in the electrical wirings. We can also use, it means don't think that we use graphene only as a coating to prevent the frost. We also use it, okay, as electrical wires. We can use it in the cell phones, okay. In many places we can use it to improve the conductivity, okay, or increase the strength of that particular structure by reducing the size. Is it clear? For example, we have cell phones. In the cell phones, we have lithium ion batteries, isn't it? In the cell phones, we have lithium ion batteries, okay. In the lithium ion batteries, normally we use graphite as an electrode. Is it clear? We use graphite as an electrode. Instead of graphite, if we use graphene, then what happens? That lithium ion battery will be slim but strong. Is it clear? We get more durable and sleek cell phones. In future, our cell phones are going to be like this. So because of all these things, I hope it is clear. I repeat, in our cell phones, laptops, like tablets and all, we use lithium ion batteries. The lithium ion batteries use graphite as one of the electrodes, but graphite is heavy. Instead of graphite, if we use graphene, then what happens? The cell phone can be slim and sleek. At the same time, it can be much stronger. So like that, we have multiple functions of this graphene. I hope it is clear. And the right answer is 3B, 2 and 3 only. Clear for all of you? Can we move on? Right. Moving on to the, no, graphene is not used in the DNA analysis. Moving on to the next topic. DNA, this is next topic, don't get confused. Okay, moving on to the fourth topic, that is DNA analysis in the newborns. Okay, outperforms the standard tests. Is it clear? So, what is DNA? DNA is our genetic material, isn't it? Second part of the fourth statement. Stronger than the steel, conductive. Yes, it is conductive than copper. It is more conductive than copper. So that is the reason why we can use it in the electrical wires instead of the copper. Normally we use copper in the electrical wires. Why do we use copper in electrical wires? Because copper is more conductive. But there is a problem with using copper in the electrical wires. That is, whenever you get a strong wind, okay, the electrical wire can break. You need more maintenance, isn't it? Weaker. Copper is not a strong metal, right? So if we replace graphene, that is this carbon nanotubes, we replace this carbon nanotubes CNTs. Okay, CNT is nothing but a rolled up sheet of graphene. Is it clear? So, like just like graphene, carbon nanotube is also highly conductive. So, graphene and therefore carbon nanotubes both are stronger than the steel and conductive than the copper. Yes, correct. Okay. Now moving on to the fourth topic. Can we proceed? DNA analysis in the newborns outperforms the standard tests. This is a news article from the 27th October 2024 from the Hindu. Is it clear? Saurabh, if you make graphene like a wire, then only we call it as carbon nanotube, isn't it? Graphene wire is nothing but carbon nano. The rolled up sheet of graphene is nothing but carbon nanotube, right? I hope it is clear, Saurabh. Right. What is DNA? DNA is deoxyribonucleic acid. DNA is deoxyribonucleic acid, and it is our genetic material. It is our genetic material. Okay, and it is made up of nucleotides. Just like protein is made up of amino acids, just like protein is a chain of amino acids, DNA is a chain of nucleotides. Each ball is like adenine, thymine, guanine, cytosine. Okay, like that. Is it clear? Adenine, thymine, guanine, cytosine. I hope it is clear. So, protein is a chain of amino acids, DNA is a chain of nucleotides. We have only 4 nucleotides, adenine, thymine, guanine, cytosine. Okay, and we all have a different sequence of DNA. We all differ in the sequence, that is order of nucleotides. Is it clear? What makes you different from me? Why do you and me look different? Because the order of these letters is different in both of us. Is it clear? Okay. Then, can we diagnose diseases, genetic diseases? Can we diagnose genetic diseases based on this DNA sequencing? Can we diagnose genetic diseases based on the DNA sequencing? For example, okay, you normally have, okay, C here, but if the C is replaced by T, then this is a mutation, and the mutation cause diseases, isn't it? I repeat, we can diagnose the genetic diseases by sequencing the DNA, by reading the DNA sequence. Because when the sequence of nucleotides in a gene changes, that is nothing but mutation. What is a mutation? Change in the gene sequence. And mutations will cause the diseases. Mutations will cause diseases. Okay. So, there is one idea that for all the newborn babies, what we have to do? We have to read the complete nucleotide sequences of their DNA. Is it clear? For the newborn babies, okay, isolate the DNA and read the nucleotide sequence. Is it clear? From the newborn individuals, we have to isolate the DNA and read the nucleotide sequence. For whom? For all the newborn babies. If we do like this, what is the advantage? We can diagnose those diseases that cannot be diagnosed by regular methods. That means the methods that we are already using. Okay. If you observe, whenever a baby is just born, the pediatrician will do some testings, like physical examination, okay, or some blood test, like basic tests will be done. Why? To know whether the baby is having any genetic diseases. Because some diseases will express in the later part of the life, when the baby is 20-year-old, 40-year-old, 60-year-old, like that. Is it clear? For example, do you agree that cancer is a hereditary disease? I mean, if you have a family history of cancers, there is an increased chance of cancer in that person if there is a family history. Okay. So, if you have a cancer causing gene, you can diagnose it the day the baby is born. Is it clear? I mean, at the age of 50, whether this baby will get cancer or not, you can tell the day the baby is born. I hope it is clear. Yes, by sequencing the DNA. Okay. I repeat, the newborn baby, isolate the DNA from that baby, read the nucleotide sequence, okay, and we can identify the mistakes in that DNA sequence. Mistakes in the DNA sequence are nothing but mutations. Mutations cause diseases. For example, at the age of 50 years, whether this individual will get cancer or not, going to get cancer or not, you can tell the day the baby is born. This is the advantage of newborn screening. We are using the word screening. Why are we using the word screening? Okay. Is there any difference between screening and the diagnostic test? Is there any difference between screening and diagnostic test? We call it as diagnostic test if only the patient will get tested. If the patient gets tested, we call it as diagnostic test. We call it as screening if everybody gets tested. Is it clear? It means it is not that this newborn baby is looking abnormal, so we did the DNA sequencing. Not like that. The baby looks perfectly alright. Still we do the DNA sequencing. In that case, we cannot call it as a diagnostic test, but we call it as a screening test. Okay. Around 8 topics we are discussing today. The duration of this class is 2 hours. We'll be closing the class at or slightly before 10 o'clock. Okay, I hope it is clear. Yes, only the patient gets tested, that is called, or you have a doubt, you have some symptoms, then you get the test, then it is called diagnostic test. No symptom, nothing, perfectly alright, still every newborn baby will get tested, then we call it as screening. Okay, I hope you got some background. So now let us give it a reading. The early results from a study called as Guardian. What is Guardian? Is it the name of an institution or an organization or a project work? What it is? It is the name of a study. It is the name of a study. Is it clear? In this study, they have taken 4,000 newborn babies. Is it clear? How many newborn babies they have taken? Mumtaz Sheikh, we are going to discuss that. Is it clear? In that study, 4,000 babies were taken. Okay. Out of 4,000, how many babies are diagnosed with some serious genetic diseases? How many babies are diagnosed with serious health conditions? 120. Is it clear? Okay. These 120 babies are diagnosed with genetic conditions by what method? DNA sequencing, isn't it? The serious genetic diseases in 120 babies were diagnosed using the DNA sequencing. Clear for all of you? Right. But if you don't use DNA sequencing, if you go by the same old methods, the other methods, traditional methods of screening if you are using, then only 10 out of 4,000 babies were diagnosed with genetic diseases. It means 110 babies, they had diseases, but we failed in diagnosing them. Is the problem understood? They've taken 4,000 babies, did the DNA sequencing and found that 120 babies have serious health conditions. But if you don't do the DNA testing, if you follow the routine traditional methods, only 10 of these babies were diagnosed with the genetic diseases. It means 110 babies had diseases, but they were not diagnosed. Is it so that they could not take any precautionary steps? Did you understand? They have to take the action only after the disease comes out. The disease starts expressing itself. Is it clear? Therefore, this technology has a potential to detect thousands of genetic diseases, far more than approximately 60 disorders that standard newborn screening now detects. Is it clear? Okay. Is it clear? Long-term diseases, any genetic disease, any disease caused by genes. Okay, Harsh, clear? Done. So now tell me, the standard newborn screening is better or the DNA sequencing of every newborn is better in diagnosing the diseases before they appear? Standard test means we are not discussing the list of it, but many blood tests are blood test, urine test, like that, not the DNA test. Okay, blood test of the newborn baby, urine test of the newborn baby, physical examination of the newborn baby, checking for the symptoms, whether the baby is properly crying or not, whether the baby is responding when you touch the baby, something like that. Okay, standard newborn screening cannot detect all the diseases. DNA sequencing can detect all the diseases. So DNA screening is better than the standard newborn screening procedures that are currently being utilized. Clear for all of you? Right. So that is all about that article. Now answer this question, fourth question. Okay. If you do not understand one or two statements, don't worry. I will explain the statements. Once you guess your answer, I will explain the statements. Genome is a haploid genetic content of a cell. Baby-seq is a large-scale study to measure the utility of genome sequencing in the newborn screening. Guardian is the first randomized clinical trial by Columbia University to use genome sequencing as a method of newborn screening. I know that, okay, baby-seq, genome, these words are new for some of you. At least I did not explain those words. What is genome? What is baby-seq? I did not tell you. Take it like an additional information, extra information. Just guess the answer. Fourth question. The right answer is A. Okay. Now, let us try to first understand what is the genome. Let us try to understand what is genome. Clear? Use the word once. Genome. Do you know how many chromosomes we have in each cell of our body? How many chromosomes we have in each cell of our body? 46 chromosomes. In this, we get 23 from our mother, and the other 23 chromosomes from our father. Is that clear? So we have to say 23 pairs of chromosomes. Is it clear? We have 23 pairs of chromosomes. Each chromosome we have in 2 copies. For example, first chromosome, 1 copy comes from the mother, 1 copy comes from the father. Second chromosome, 1 copy comes from the mother, 1 copy comes from the father. 23rd chromosome, 1 copy from the mother, 1 copy from the father. Each chromosome, 1 copy from the mother, 1 copy from the father. So we say 23 pairs of chromosomes. Is it clear? Then in this, one set is called as genome. We have 2 sets, isn't it? We have 2 sets. And one set is called as genome. Is that clear? So one genome is equal to how many chromosomes? We have two sets, one set of 23 chromosomes from the mother, one set of 23 chromosomes from the mother, one set of 23 chromosomes from the father. So we have total two sets of 23 chromosomes. And genome is only one set. That means one genome is equal to how many chromosomes? Perfect. Very good. 23 chromosomes is one genome. Okay. So genome can be defined as, just 1 second. Genome can be defined as the haploid. Haploid means half. The word haploid means half. Haploid genetic content of a cell. Haploid genetic content of a cell. That is half the total genetic content of a cell. Total you have 46 chromosomes. Half of it is called as genome. Fine. If that is clear, tell me how many genomes you have in one cell. How many genomes in one cell? One genome is 23 chromosomes. So how many genomes we have in one cell? Yes, we have two genomes. We have two genomes in one cell. One genome from the mother, one genome from the father. Is it clear? One genome, one set of 23 chromosomes from the mother, one set of 23 chromosomes from the father. So genome is the haploid genetic content of the cell. Haploid means half. Genome is half the genetic content of a cell. So first statement is correct. Second statement is just rearranged. I just rearranged it. So both of them are wrong. It means the first effort to sequence the newborn baby's DNA is baby-seq. This is the recent one. This is the recent one. This is in the news on 27th October 2024. Is it clear? This is from October 2024 news. Okay. But baby-seq is I think 2018 or 2019. I don't exactly remember the year. But it is an older, both are similar. Both are studies of genome sequencing in the newborn babies to screen them for the genetic diseases. Is it clear? Newborn screening using DNA sequencing. Newborn screening using the genome sequencing. What are the two studies? One study is baby-seq, the other one is guardian. The first randomized clinical trial, they tried in the clinics first time, that is correct for baby-seq. Large scale study is this guardian study. It's a large scale, 4,000 babies sequenced. Okay, but it is not the first such effort. I hope it is clear. So both the statements are wrong. So the correct answer is, which of the following statements are correct? One only is the right answer. I hope it is clear for everybody. Yes? Okay. Moving on to the next topic, the fifth topic, and I will be discussing total 8 topics in today's class. The class is expected to approximately end at 10 p.m. Okay. The next one is a simple topic. It is from Indian Express, 12th November 2024. The significance of Lignosat. Use the name once. What is the name of the satellite? Lignosat. What is the speciality of this satellite? It is a wooden satellite. Wooden satellite launched into the space. Lignosat is the world's first wooden paneled satellite. The satellite is paneled, covered by a wooden panel. This is launched into the space, marking the first step towards renewable material in the space exploration. So this is the first time we are using a renewable material like wood, okay, in the manufacture of spacecrafts. Is it clear? First time we are using a renewable material or environment friendly material in the manufacture of the satellites. This is a Japanese satellite. It will orbit the Earth for 6 months. Okay, and it will be launched from the International Space Station. I mean, it will first go to International Space Station. From the International Space Station, it will be launched. First, it will be launched from the Earth. Then it will reach the ISS. From the ISS, it will come out and will orbit the Earth for approximately 6 months. At least expected to orbit for 6 months. Is it clear? It is a small sized cube shaped, like a cube, okay, device measuring 10 centimeters on each side, weighing 900 grams. Okay, I hope you all are following. It features panels made of Magnolia wood, which is crafted using the traditional Japanese woodworking. Do you know, did you see any videos regarding this Japanese woodworking? They don't use any nails in the woodworking. Same way, they don't use any glue like Fevicol or any screws. Okay, they will just do the woodworking like a jigsaw puzzle. Like a puzzle, the two pieces of wood will join with each other. Okay, so if you are interested, you can watch some videos related to the Japanese woodworking. Is it clear? Like we do Lego blocks, like that. Okay, without nails, without screws, without glue. Is it clear? So it is made, Lignosat is made using such technique only. Okay, Lignosat will monitor the wood's endurance in the space. What is the purpose? What is the aim of this Lignosat? So first one, it will test whether that wood will tolerate the space situations or not. The temperatures here will range from -100 to +100 degrees Celsius. Okay, it means 200 degrees Celsius variation is there. So can this wooden satellite tolerate such temperature extremities? This is one thing which we want to test. Okay, what are we discussing? We are discussing the target, the purpose of Lignosat. Why did we launch Lignosat? Okay, because we want to monitor whether the wood can tolerate the temperature extremities. For every 45 minutes, we will monitor how the wood is reacting to such extreme temperatures. Researchers are examining wood's resistance to the space radiation. In the space, we are getting radiation like X-rays, gamma rays, infrared, visible light, different radiations are coming. Is the wood resistant to the space radiations? Okay. Next, third one. First one, temperature extremities. Second one, resistance to the radiations. Third one, can it shield the delicate semiconductors which are inside the Lignosat? Can the wooden panels shield, protect the delicate semiconductors? Is it clear? Which type of renewable material explode? Wood, na? Okay, Lalita, we are using wood. Lignosat is made up of wood. Wood is a renewable material because plants keep growing. Okay, wood, and maybe in the future we try to use more renewable material. Presently, the renewable material we are using is wood. Is it clear? Did you understand? Okay, so first one, is it resistant? Is the wood resistant to temperature extremities? Is the wood resistant to the space radiation? Third one, can it protect the delicate semiconductors which are there inside the satellite? And also to explore sustainable building options potentially for future habitats on the moon and Mars. Fourth purpose. Is it clear? I hope you are identifying the goals, targets of the Lignosat. Okay, it means we want sustainable building options. I mean the building options which will not pollute the space. The building options which are not increasing the pollution, cost effective, environment friendly, not very polluting. Is it clear? This kind of material we want to use for future habitats. I mean, in future if you construct a home on the moon or Mars, then it should be with wood. Is it clear? Because it is sustainable. And it is also with the goal of testing timber as a sustainable alternative to metals. I mean, Presently, we are using metals like aluminium and the other metals. Is it clear? Presently, what is the commonly used material in the spacecrafts? Metals like aluminium and some other metals. But what happens? They cause pollution upon re-entry. Did you hear about space junk? Space junk or space debris? These are the small pieces, okay, man-made objects, that is like the retired satellites, okay, used up stages of the rockets, okay, failed satellites, all these will form the space debris. They behave like bullets because they're very fastly orbiting the earth. So they will behave like bullets. It means if any satellite comes on their path, they will hit the satellite and damage it. So now tell me, do we have a responsibility of decreasing the space debris? Do we have a responsibility of decreasing the space debris? Yes. Okay. We use them in the pure form and also alloys. Both are used, Harsh. Is it clear? We have to decrease the space debris. Okay. How to decrease the space debris? Okay. How to decrease the space debris? Okay. By re-entry of used up stages of rockets and retired or failed satellites. Okay, retired or failed satellites. Is it clear? That is what Lalita Prasad, I am explaining that. Pollution upon re-entry is what I am explaining right now. So listen to that. We have the problem of space junk. Lalita Prasad, we have the problem of space junk, space debris, that can speedily hit our satellites and damage them. So we have to minimize the space junk. Okay. To minimize the space junk, what we have to do? We have to bring the satellites. What satellites? Retired satellites whose lifespan is completed, like Mars orbiter mission. Okay. Use retired satellites whose lifespan is completed, or failed satellites, or the used up stages of rockets, the rocket which we used to launch the satellite. All these should be brought back into the Earth's atmosphere. That is called as re-entry. I hope the re-entry concept and purpose is clear. What is the purpose of re-entry? To minimize the space debris. Is it clear? Of course, Pavan Kalyan, correct. We can use reusable launch vehicles. That is one option. But what is the other option? What is the option to decrease the debris which is already there? Bring them back onto the earth. When you bring them back, if they are made up of metals, they will cause pollution when they are entering back into the earth's atmosphere. That is, contribute to pollution upon re-entry. Lalita Prasad, I hope it is clear. Space debris, minimize it. One way is used to use the re-entry vehicles, reusable vehicles, true. But how to reduce the space debris which is already there? Okay, bring them back. Okay, bring them back into Earth's atmosphere. When they are coming back, they will cause pollution if they are formed, if they are manufactured using the metals. Is it clear? While they are coming back, they will cause pollution if they are made up of metals. But if they are made up of wooden, if we use wooden satellites, they will not cause pollution, okay, or cause very little pollution comparatively when they are re-entering the Earth's atmosphere. It will simply wooden. So it will simply get burnt once it enters into the Earth's atmosphere due to the friction, okay. The Earth's atmosphere is thick. So while any spacecraft is entering into the Earth's atmosphere, it will create huge friction. The friction will create heat. Due to that heat, the wooden piece will be burnt. It will cause less pollution or no pollution. So that is the other purpose of it. I hope all the aims or purposes of the Lignosat clear. It is a Japanese satellite. Clear till now? Yes? Now, do the question, the fifth question. Consider the following statements. Which of the above is not a goal of Lignosat? Monitoring wood's endurance in the space, wood's resistance to space radiation, testing the potential of the wood to shield the semiconductor, potential means capacity, reduce the pollution caused by aluminum and other metals upon re-entry of the spacecraft. Yes, D is the right answer because all of them are correct. And the question is not a goal. None of the above is not a goal because all of them are the goals. That's it. Okay. Now, moving on to the next question. Yes. A mini protein carries radiation dose directly to the tumors. Okay, let us try to understand. Already we have seen about cancer. Tell me some commonly used methods of cancer treatment. Some commonly used methods of the cancer treatment. Chemotherapy. Chemotherapy. It means using some chemical drugs. Isn't it? Using some chemical drugs. I hope it is clear. Right. Second one, radiotherapy. Radiotherapy. In this we use radiation. Can you tell me what rays are used? UV rays, X-rays, light rays, visible light, IR. Which radiation? We use gamma rays. Okay, gamma rays we use. These are the highest frequency radiations in the electromagnetic spectrum. Is it clear? After the X-rays we get the gamma rays. They will burn the cancer cells. They can burn the cancer cells. Is that clear? Next, okay, you can also do surgery. You can also remove the cancer by a surgery. These are the commonly used methods, isn't it? These are the three common or regularly used methods. Is it clear? Then, okay. Now, this is one piece of information. Now, I'll give you another small piece of information. Okay. This is an overview of our immune system. Is it clear? Some information about our immune system. We have two types of immune system. Can anybody tell me what are the two types of immune system we have? One is antigen, antibody mediated immune system. The second one is cell mediated immune system. Is it clear? Okay. So immune system, we have two types of immune, not primary, secondary. We have antigen and antibody mediated immune system and cell mediated immune system. Is that clear? Okay. And antigen and antibody means usually, okay, it will destroy foreign antigens like bacteria, etc. I mean pathogens, disease causing agents. Okay, is that clear? I'm explaining, Lalita. Okay, actually I'm explaining each and every word. Just continue to listen. Okay, first of all, is the antigen and antibody mediated immune system clear? Lalita Prasad, first answer this question. Okay, if a pathogen enters into our body, Okay, what is a pathogen? Anything that can cause disease. It can be a virus, it can be a bacteria, it can be a fungus, anything that can cause disease is called as a pathogen. Pathos means disease, gen means causing it. Okay. If a pathogen is coming from outside, say H5N1 is coming, coronavirus is coming, is it an antigen or an antibody? What is that bacterium? Is that bacterium an antigen or an antibody? Yes, that is an antigen. And is this antigen made in our body or coming from outside? Is this antigen made in our body or coming from outside? No, Harshu, it is not antibody, it is antigen. It is an antigen coming from outside. Means it is a foreign antigen. Is it clear? Then a cell called B cells. B cells produce antibodies to kill or destroy that foreign antigens. Is it clear? To destroy that pathogen, to destroy that virus or bacteria, some cells called B cells produce the antibodies. Tell me, what cells of our immune system produce antibodies for foreign antigens? What cells of the immune system produce antibodies for foreign antigens? B cells. So, can I compare this type of immune system as border security force? Can we compare this with the border security force? Our army stays at the borders. Why do they stay at the borders? Because if anybody is coming from outside, to target them, to target the enemies coming from outside. Is it clear? Difference between an antigen and an antibody. In fact, I told you what is the difference between antigen and antibody. Antigen is something which is coming from outside. That bacteria, virus, these are called antigens. Antibody is a defense mechanism. It is our body's defense mechanism to destroy that antigen. Is it clear? Okay. Then, so can I call this antigen and antibody immune system as a border security force? Just for understanding. Don't write this in the exam, please. Okay. So we are comparing like this. The second type of immune system is, okay, the cell-mediated immune system. Okay. It is not targeting the foreign. It will target altered self, okay, cells. Altered self cells. Altered self cells means, okay, my cells only. They are not coming from outside, not foreign. Did you understand? Sorry, self. Okay. Self cells means what? They are not foreign. They are my cells only, but altered. Okay. So, a funny sentence to better understand this is like मेरा है पर तेरा है, मेरा ही है. Okay, self cells only, मेरा है पर तेरा है, altered. What is the example for it? Example for altered self cell is cancer cells. Tell me, for a cancer patient, cancer cells are self cells or foreign cells? For a cancer patient, cancer cells are self cells or the foreign cells? They are self cells only. Is it clear? But are they normal or altered because of a disease? Are the cancer cells normal or altered because of this condition? They are altered. So we call them as altered self cells. So now answer these two questions. Normally, should our immune system protect our own cells or damage our own cells? I mean, my immune system should protect my cells or damage my cells? My cells. My immune system should protect my cells or damage my cells? Protect. Okay. If they are self-antigens, protect. Then answer this question. If they are altered self, I mean mera hai par teda hai, if they are altered self cells, then protector damage. For example, cancer. Cancer is my cell only, but it is an altered cell, it's a modified self cell. Should we protect it or destroy it? Everybody please. Destroy. Okay. Who will destroy? T cells will destroy, not B cells. T cells will destroy. Is it clear? Okay. So can I compare T cells with internal security? Can I compare T cells with it means if an enemy is generated from inside the body, T cells will destroy. If an enemy is coming from outside the body, then B cells will destroy. How the B cells will destroy? By producing antibodies. What are antibodies? These are the defense molecules that will destroy the foreign antigens. Is it clear? So that is how we have two different types of immune system. Okay. Similar samarth, mutated, altered self cells are mutated cells. Cancers will definitely have some mutations. Cancers are caused by mutations. So altered self cells are like mutated self cells. My cells only, but modified. They are not healthy, they are diseased. So I have to destroy them. Who will destroy? T cells. So now finally answer this question. B cells are for, I mean if you get a bacterial infection, okay, tuberculosis or something, which immune system will protect you? You can use shortcuts in your answers. If you get a bacterial infection like tuberculosis, which immune system will protect you? B cells or antigen and antibody mediated immune system. B cell is not the name of an immune system. B cell is the name of a cell. Is it clear? Okay. And if you get cancer, which immune system will protect you? If you get cancer, which immune system will protect you? Yes, if you get cancer, which immune system will protect you? Cell mediated immune system. What cells will kill the cancers? No, Ayushi, antigen and antibody mediated immune system will target the foreign antigens. Cancer is not coming from outside. Okay, cancer is destroyed by cell mediated immune system, T cells. Is it clear? Not B cells. T cells kill the cancers, not B cells. Is it clear? Okay, now let us first read this article now. This is all the background information. I have given two pieces of background information. First one is what are the regularly used methods of cancer treatment. Second one, an overview of the immune system. With this background information, let us read this new article which is published in the Hindu newspaper on 27th October 2024. Researchers have shown that for the first time it is possible for a specially designed mini protein. A specially designed mini protein will deliver a radiation dose directly to cancer cells, not affecting the healthy cells. How? Look at here. Okay, let us assume this is a cancer cell. Okay, cancer cell. On the cancer cell, there is one protein called as, nectin-4. There is one protein called as nectin-4. Clear? There is one protein called nectin-4. Where is it? Tell me, where is nectin-4 protein? On the cancer cells, right? Nectin-4 protein is on the cancer cell. Then what is this? This is a mini protein, not nectin-4. That is called mini protein. Is it clear? We are calling this as mini protein. And this is nectin-4. Okay? Mini protein is carrying a radiation, I mean some gamma rays. Is it clear? What protein is delivering or carrying the radiations to kill the cancer cells? Everybody? What protein is delivering or carrying a radiation dose to the cancer cells to kill the cancer cells? Protein is a structure made up of amino acids, Lalita Prasad. And we have so many proteins like muscle protein, hair protein, keratin, milk also we have protein like casein protein, hair will have keratin protein. Proteins are the chains of amino acids. I hope Lalita Prasad it is clear. Okay. Wait, I still did not ask you the question. Okay. So, mini protein will carry. Mini protein will bind to what? Nectin 4. Where is nectin? On the cancer cells. Do we have nectin on the healthy cells? I mean non-cancerous cells? Yes? No. So, now tell me, can this mini protein deliver the radiation to the cancer cells? Can the mini protein deliver the radiation to the cancer cells? Yes. No, nectin 4 is not mini protein. Please observe it. This is nectin 4. Look at here. This is nectin 4. It is there on the cancer cell. This is, look at here, this is the mini protein which we are sending. Is it clear? Why are we sending? For therapy. Mini protein we are sending. Nectin 4 is already there on the cancer cells. But they both are like lock and key. Is that clear? They both are like lock and key. Mini protein can bind to nectin 4. So now tell me, mini protein is carrying a dose of radiation. Is it clear? So everybody answer this question. Can the mini protein deliver the radiation to cancer cells? Can the mini protein deliver the radiation to cancer cells and kill the cancer cells? Yes. Can the mini protein deliver the radiation and kill the healthy cells? Can the mini protein deliver the radiation and kill the healthy cells? No, because healthy cells do not have the nectin 4 to which the mini protein will bind. Is it clear? Healthy cells do not have the nectin 4 to which the mini protein binds. So, mini protein cannot bind to healthy cells. So, no radiation given to healthy cells. But it can bind to cancer cells. So, radiation is specifically given to cancer cells. Is this what we want in the cancer treatment or not? What is the final ultimate purpose of the cancer treatment? Kill the cancer cells while protecting the healthy cells, isn't it? Kill the cancer cells while protecting the healthy cells. That is the advantage. Can we call this mini protein as a radiopharmaceutical protein? Radio because it is carrying the radiation. Pharmaceutical because it is used in the therapy. This is like a drug. We are sending it like a drug. Is it clear? It is a radiopharmaceutical mini protein. That's it. The topic is completed. Answer this question. Consider the following. Okay. Radiopharmaceutical protein carrying the radiation dose directly to the cancer cells. Actually, what are these statements? Which of the above is not a method of cancer treatment? Okay, that is the question. Read the statements. Radiopharmaceutical protein carrying the radiation dose directly or specifically to cancer cells. Nanoparticles for targeted drug delivery. Like for example, this is a cancer cell. On this you have a receptor. And it is a nanoparticle. And you kept, okay, cancer drug inside. Okay, targeted drug delivery, where the nanoparticles are delivering the drug directly to the cancer, similar to that mini protein. Mini protein will carry the radiation. Nanoparticles can carry the chemical drug, chemical drug. Okay, modifying the T cells of the immune system to specifically target the cancer cells. Modifying the B cells of the immune system to specifically target the cancer cells. Which of the above is not a method of cancer treatment? Yes, answer the question. All of you, some of you have given the answer A, C, A, D, all answers have come. But the right answer is A, because this is correct. The article is all about the first statement, isn't it? Okay, the news article we discussed is about the first statement. Radiopharmaceutical protein carrying radiation dose directly to the cancer cells. Yes, correct. Can the nanoparticles carry the chemical drugs in a targeted way to the cancer cells? Yes, like this. Correct. Okay. Then, if you remember in this slide, I explained you that T cells are to kill the cancer cells. B cells are to kill the foreign antigens. B cells are our border security force. T cells are our internal security. So fourth one is the wrong statement. Okay, which is not a method of cancer treatment. 4 is not a method of cancer treatment. Is it clear? Then moving on to the next topic. Okay, the last but one topic. Inactivated COVID-19 vaccine CoronaVac, okay, associated with TTP. What is TTP? Let us try to understand it. I tried to explain TTP in 6 steps procedure. Follow the 6-step procedure. I will explain the 6 steps one after the other. Any step is not clear, you can say repeat 1, repeat 2, like that. Okay. Step 1. Okay. Immune cross reactivity between vaccine components or SARS-CoV-2 virus and ADAMTS113. Okay, no, this is not reassortant. That is H5N1. This is a different story. Don't mix both. Okay, right. Okay, first tell me one small information. What is there in the vaccine? Vaccine contains antigen or antibody. For example, coronavirus, we have taken vaccines, polio vaccine. We take lot of vaccines, right? Do vaccines have antigens or antibodies? Antigen. And we produce antibodies, isn't it? We produce antibodies. Clear? Yes. So we send it either in the small dose or inactivated or killed, or only one part of the subunit vaccines we call. Only one part of the virus, like that we inject. This is something like, the concept of vaccine is like digging the well before you feel thirsty. Tell me, do you have enough time to dig the well after you started feeling thirsty? You're feeling very thirsty. Can you leisurely dig the well? Or somebody already started the war on your country. Can you now start manufacturing the weapons? Inactivated means inactivated only, Lalita Prasad. We inactivate it and inject. Okay. Is that clear? No. So we have to dig the well before we feel thirsty. Manufacture the weapons before the war is declared. What are the weapons here? Weapons are antibodies. Should we produce antibodies before you get infection or after? After you get polio, you have to produce polio antibodies, or produce them before you get polio? Before. But how do we produce antibodies before you get polio? Because antibody production is a reaction to antigen. Is it clear? Antibodies are produced when they get exposed to the antigen. Antigen entry is action. Antibody production is reaction. So how can you produce polio antibodies before you get polio? By vaccination. We inactivate the polio virus and inject it, or we kill the polio virus and inject it, so that our immune system is exposed to the antigen before you actually get the infection, so that we produce antibodies. Now you are ready to fight with the pathogen. If the pathogen really comes, you can easily fight with the pathogen. You will not get the disease. Even if polio enters into your body, okay, the antibodies will fight with that. It cannot cause the disease. Is it clear? This is the story of the vaccine. So, do you agree that, vaccine will have coronavirus, okay, and the real infection also have coronavirus, isn't it? Which one? The SARS-CoV-2. Isn't it SARS coronavirus 2 that causes COVID-19? Do you agree with this? Real infection is also having the virus, antigen. Vaccine also contains antigen. Is it clear? No, it will not get activated. If it gets activated, you will get polio. Okay, if the polio virus gets activated in your body, you will get polio. So the polio virus remains inactive, but it is an antigen, right? So our body will produce the antibodies. Our immune system will produce the antibodies. That's it. Okay? Right. Okay. So we'll discuss that towards the end of the class, Bahadur. I hope it is clear for everybody. Because I'm falling short of time, I have to complete it before 10 p.m. Right? Okay. So if you inject inactivated virus, it will remain inactive only. But inactive virus can cause the production of antibodies. Now antibodies are ready. If the real virus comes, these antibodies can destroy the real virus. So even if the real virus comes, you will not suffer from the disease. That is the concept of vaccine. So do you agree? Vaccine and the real infection both have the same antigen or the same pathogen, yes or no? Vaccine is also, polio virus will be there in the polio vaccine. Coronavirus is there in the coronavirus vaccine. Hepatitis virus is there in the hepatitis vaccine. I hope you got it. Right. So, this antigen, the antigen either in the vaccine or the virus is having cross reactivity with another antigen we have, okay, in our body called as ADAMTS13, okay, ADAMTS13, let us call it as. Is it clear? ADAMTS13, okay. What is this? This is a human protein. This is our protein, not the virus. Is it clear? Okay. Means what I am trying to explain is, okay, the SARS-CoV-2 antigen, either in the vaccine or in the real infection, is similar to ADAMTS13 protein. What is this? This is not viral protein. This is a human protein. Is that clear? Yes, we are inserting virus. Just like we say, we use diamond only to cut the diamond. Like that, to protect from the virus, we inject virus. Is it clear, Mumtaz? Okay. Yes, I'm Lalita Prasad. I'm explaining what is cross reactivity. I circled it and I'm explaining that word only. Is it clear? Okay, I'm explaining the concept of cross reactivity. What is cross reactivity? Cross reactivity is, look at here, we are getting the coronavirus antigen. So Lalita Prasad, tell me, how are we getting coronavirus antigen? Either by real infection or by vaccine. Is that clear? Okay. The SARS-CoV-2 antigen. Is that clear? Now, okay. Then what happens? We produce antibodies for SARS-CoV-2 antigen. Step 1, step 2. Is that clear? Step 1, SARS-CoV-2 antigen entered our body. I repeat, step 1, SARS-CoV-2 antigen entered our body. How? Either by vaccine or by real infection. Step 2, our immune system will produce antibodies for SARS-CoV-2. I hope everybody is understanding. Okay. Step 3, the point number 3, there is a human protein which is accidentally similar to the SARS-CoV-2 antigen. Is it clear? Structurally, that is similar to the SARS-CoV-2 antigen. Is that clear? So, fourth step, these antibodies will destroy that ADAMTS13 also. How? By mistake. That is not the intended action. That is by mistake. I hope all of you are understanding. Step 1, SARS-CoV-2 antigen entered our body. How? Either by vaccine or by real infection. Step 2, our immune system, that is B cells, produced antibodies for it. Okay. Point number 3, there is one human protein called ADAMTS13 which is similar to the coronavirus antigen. Why similar? Just coincidence. There is no reason for it. Coincidence. Coincidentally, it is a human protein which is similar to the SARS-CoV-2 antigen. Is that clear? So, by mistake, the coronavirus antibodies will destroy that protein also. Is it clear? Everybody? Should we destroy that or not? Should we destroy the ADAMTS13 protein? Should we destroy the ADAMTS13 protein? Should we destroy? Should we destroy? Lalita Prasad? No, we should not destroy because it is a human protein. We should not destroy because it is a human protein. But do we destroy? Are we destroying that protein? Are we destroying that protein? Yes. Okay. Why? By mistake. Why we are destroying these proteins? Because coincidentally this protein is similar to the coronavirus antigen. I hope it is clear. Coincidentally that protein is similar to the coronavirus antigen. Done? That is called cross reactivity. What is cross reactivity? Okay. The SARS-CoV-2 antibodies are reacting with ADAMTS13 protein. That is called, okay, cross reactivity. Is that clear? How they are destroying? They are destroying because they are coronavirus antibodies. But this ADAMTS protein is looking like coronavirus. So they will think that innocently, they will think that this is coronavirus and destroy it. Lalita Prasad, clear? Coronavirus, they think that it is coronavirus and destroy it. That mechanism is called cross reactivity. Two antigens are similar. So the antibody by mistake reacting with the other antigen is called cross reactivity. Yes, clear for all of you? Okay. So, I said I am explaining this in steps. So, what is the step 1? Step 1 I explained in 4 steps. Did you understand? Step 1 I explained in 4 steps. Immune cross-reactivity between vaccine components or SARS-CoV-2 and Adam TS13. Okay, how did I explain? The virus enters vaccine or the real infection. Then what happens? The Adam TS13, which is a human protein, is similar, coincidentally similar to the coronavirus. Then the antibodies will by mistake assume that it is coronavirus and destroy that. That is called cross-reactivity. Two antigens are similar. Then the antibody of the first antigen by mistake reacts with the antigen of the other antigen. That is called cross-reactivity. I repeat, two antigens are similar. Antibody for the first antigen by mistake reacts with the second antigen. That is called cross-reactivity. I hope it is clear. Right. Step 2. Okay. Generation of Adam TS13 antibodies. Okay, this leads to deficiency of Adam TS13. Why? Because this is destroyed by the antibodies. Destroyed by antibodies? Is that clear? Real infection means real infection only when you get COVID-19, not the vaccine. When you are getting the virus naturally, not by vaccination. Is that clear? Okay. So, like either you got the infection, you got COVID-19 or you got vaccinated, step 1. Step 2, antibodies for coronavirus produced, but they also destroyed Adam TS13. So what happens? Deficiency of Adam TS13. Lalita Prasad, why Adam TS13 is deficiency is occurring? Because that is being destroyed by that antibody. Is it clear? True, Samarth, it can also happen, but there is one rule that we will not produce antibodies for self-antigens. Do you remember that? We have a rule that we do not produce antibodies for self-antigens. So, we don't produce this antibody before the infection comes. Samarth, I hope it is clear. Okay, right. So, step 4. Blood clots are formed. Why blood clots are formed? Because, this Adam TS13, what is the role of Adam TS13? Adam TS13, which is dying, its role is prevent blood clots. Is it clear? Tell me, what is the natural function of Adam TS13, which is accidentally dying because of this vaccination or COVID-19 infection? Everybody? What is the biological role of Adam TS13? Prevent the blood clot. But now do you have enough Adam TS13? Now do you have enough Adam TS13? No, Adam TS13 is destroyed. So what happens? Blood clots. The blood clots is called thrombosis. What is the meaning of thrombosis? Thrombosis means blood clots. Clear? Thrombosis means blood clots. Okay. Now, formation of blood clots consumes platelets. Did you hear that in the blood clots we have blood platelets? Dengue virus, dengue infection also, platelet count decreases. So you will get bleeding, internal bleeding in dengue infection, isn't it? I hope you all know that blood platelets will help in the blood clotting process. Is it clear? I mean, all the blood platelets will clump together. That is nothing but the blood clotting. Is it clear? The blood platelets will clump together like this. That is nothing but the blood clot. Is it clear? So this thrombosis, blood clots will consume the platelets that lead to decrease in the platelet count. That leads to decrease in the platelet count, which is called thrombocytopenia. What is a thrombocytopenia? Simple, decrease in the platelet count. Is it clear? Okay. I hope thrombosis is clear. Thrombocytopenia is clear. What is thrombocytopenia? Decrease in the platelet count. Okay. Next, decreased platelets leads to internal bleeding underneath the skin. We need platelets for blood clotting, but all the platelets are consumed in that thrombosis. Is it clear? So now we do not have the platelets, enough platelets in the blood. So let us suppose somebody just hit me like this. Then what happens? Internal bleeding. That blood will not clot. So I will get purple color patch on the skin. I repeat, somebody hit me like this. I got internal bleeding because some of my blood vessels ruptured. Normally my blood would have clotted, but now my blood is not clotting. Why the blood is not clotting? Because of the thrombocytopenia, that is decrease in the platelet count. So because of the bleeding underneath the skin, I will get purple bruises, that is called purpura. Together it is called thrombotic thrombocytopenia purpura. Is it clear? Thrombotic thrombocytopenic purpura. Clear? Thrombotic is blood clots. Blood clots. The blood clots will consume the platelets. So thrombocytopenia means decrease in the platelets. Decrease in the platelets. Purpura means purple patches. Purpura means purple patches. I hope it is clear for all of you. Right. Okay. Right. I'm happy that Lalita Prasad understood. Okay, yes, 3 and 4. I will repeat again. I'll repeat the total story once again. Okay, right. But for that, Mumtaz, first tell me, what is the human protein which we need for the blood clotting? Name the human protein which we need for the blood clotting. Mumtaz and everybody, please. Other vaccines also show similar effect, Samarth. Your point is correct. Other vaccines also show similar effect. Okay? Yes. Now, okay, we need this for blood clotting. So, step number 1, you have taken the vaccine, or you got COVID-19 disease, means coronavirus entered. Now, coronavirus is structurally similar to ADAMTS13. So, we produce antibodies. Antibodies will destroy the coronavirus. Very good, great job, keep it up. But they will also destroy the ADAMTS13 protein. Is it clear? Because of that, what happens? ADAMTS13 protein deficiency. Okay. Because of this, what happens? Okay. It will lead to many blood clots. It will prevent the blood clotting. So, it will lead to many blood clots. That is thrombosis. Many blood clots. That is thrombosis. Okay. And these formation of blood clots will utilize the platelets. So, platelets are used for this blood clots, for this thrombosis. Now, the platelet count decreased in the blood. Platelet count in the blood decreased. So, because of the decreased platelets, okay, what happens? Okay, we will get lack of clotting. So, it means I'll just repeat. ADAMTS13 prevent clotting. Prevent clotting. Is it clear? Platelets cause clotting. Is it clear? Now, ADAMTS13 is decreasing. Okay, because antibodies, the coronavirus antibodies, okay, are destroying that. So, its function is prevent clotting. Now, this is not there to prevent. So it will lead to clotting. That is called thrombosis. That is called thrombosis. Is it clear? Okay. Then the thrombosis, the blood clotlets, okay, will utilize the platelets. Platelets are used up, completed. Is it clear? Platelets are completed. So they will cause clotting. So it means no blood clotting. No blood clotting. Is it clear? Underneath my skin, blood is not clotting. So I will get bleeding underneath the skin. That is called purpura, purple color bruises. Why? For example, somebody hits me. Okay. Internal blood vessels ruptured. Bleeding occurred. But clotting is not occurring because platelets are not there. So bleeding underneath the skin will lead to purple patches on the skin. That is called purpura. Is it clear? Okay. Lack of necessary clots due to excess formation of the clots. Perfect. If blood clots happen inside due to deficiency of that specific hormone leading to thrombosis, doesn't it block the flow of the blood? Yes, it will block the flow of the blood. That is thrombocytopenic purpura. It can cause death of the person also. These excess blood clots because of the deficiency of ADAMTS 13 can lead to the death of the person. Is it clear? But this can happen either due to coronavirus infection or due to the coronavirus vaccine. Is it clear? Yes. This is the reason for the increased heart failures. Okay. But don't blame the vaccine. It will happen because of the virus also. And the vaccine will reduce the viral infections. So, actually we should not blame the vaccines. Did you understand the point? I repeat, the blood clots can lead to obstruction in the blood vessels so that it can lead to heart failure. True. It is a side effect of vaccine, but most importantly remember that the thrombocytopenic purpura can also occur because of the coronavirus also, not only vaccine. If you get COVID-19 also, this can occur. Vaccine in fact reduce the TTP cases by controlling the pandemic. If the vaccines are not there, probably we will be in the 6th wave or 7th wave. Did you understand? So more people would have died due to TTP. Did you understand? No, Samarth, we don't have it. This happens after vaccine, but this will happen. Why the people died due to COVID-19? That is also TTP. Did the people die only due to the vaccine? Did we give the vaccine for a time pass? We have given the vaccine when thousands of people are dying due to COVID-19, isn't it? Okay. So people are dying due to COVID-19. That mechanism is also TTP. Similar reasons, vaccine, what do you have in the vaccine? You have the same antigen in the vaccine. So vaccine also will cause it. So of course it is a side effect of vaccine. Agreed, true. But if vaccines are not there, more people could have died due to the COVID-19 infection. The infection also causes this. I hope it is clear. Yes, that is a rumor. Whenever we speak about anti-vaccination arguments, please remember 1947, our average life expectancy was 39 years. Today, our life expectancy is 70 years. Is it clear? This much life expectancy increased. One primary reason is vaccines. I have many reasons, but one primary reason is vaccines. Is it clear? So we should never blame the vaccines. If you blame the side effect, direct effect, that is death will be there. Did you understand? more people would have died due to thrombotic thrombocytopenic purpura due to COVID-19 than the people dying due to this TTP due to the vaccination. Is it clear? Okay. So we should not blame that. Now, okay, look at the question. Which of the following is not known to cause the thrombotic, TTP? Okay, incompletion. Of course, yes. But side effect, it means most of the people, for example, I have taken 3 doses of vaccine, but I did not get TTP. Most of us did not get TTP. 0.001% cases they got. More people would have died due to TTP if the vaccinations were not there. I hope you got it. Okay. But pros are more than the cons. Pros and cons are there for science, but the pros are more than the cons. Is it clear? At least for vaccines. I mean, if you ask me, for example, nuclear weapons, that is also science. I will definitely say the cons are more than the pros. Okay, but now vaccines, I will clearly say the pros are more than the cons. I mean, we should not generalize. Okay, that is everything will have pros and cons. No, vaccines are great. Is it clear? Nuclear weapons cause violence. There, cons are more. Here, pros are more. I hope it is clear. So I don't like the anti-vaccination drives. So just if the vaccines are not there, smallpox is 100% lethal. If you get smallpox, they used to die 100 years back. People used to die due to smallpox. Okay, is it clear? So, otherwise, just imagine, do not die, but surviving with polio. Just imagine surviving with polio. That is how advantageous the vaccines are. So, finally, do vaccines cause TTP? Yes. So, is it bad? Yes. Can it lead to sudden death? Yes. But what percent? 0.001 cases. If vaccines are not there, COVID-19 would have caused TTP in more cases. Is it clear? So it is a side effect, but we should not blame the vaccines for the side effect. I hope you got it. Okay. Which of the following is not known to cause the TTP? Okay. Pfizer BioNTech mRNA vaccine, it will cause TTP. AstraZeneca's Covishield vaccine, that is also known to cause TTP. Inactivated vaccine called CoronaVac. Actually, the news is about that. I think we already forgot the news. Okay, the news is about CoronaVac. Yes. Okay, right. Then SARS-CoV-2 infection. Yes, the actual infection also will cause TTP. So what? Okay, is not known to cause TTP, none of the above. The right answer is E. E is the correct answer. Is it clear? Actually, I planned 8 topics, but it is already 10:14. And I have another session on November 25th. I have one more similar session on November 25th. Roughly, the news articles from the same duration will be discussed in that class also. The 8th question or the 8th topic that I could not explain in this class due to the time constraint will be explained in the next session, which is there on the 25th. So that's all for today's class. Thank you so much. Good night. Take care. Still Roger, it will cause TTP. Still mRNA vaccine will cause TTP. Thank you so much for your active participation. That's all for this class. Good night. Take care.